human microbiome and how it plays a fundamental role in host immunity and host metabolism and the overall health of the human being. And how it can also, we can use it to prevent or even treat certain diseases. Our guest speaker is Dr. Ibrahim Alhifzi. He's a consultant neonatologist. He's interested in the microbiome, and he's also the vice president of the Saudi Neonatology Society. Unfortunately, Dr. Ibrahim won't be able to be with us here, but he will give the lecture over Zoom, sorry. So yeah, Dr. Ibrahim, can you hear me?
Yes.
Yeah, so...
Can I start?
Yes. You can go ahead and start.
Alhamdulillah Rabbil Alamin, salatu wassalamu ala Rasulullah. Good afternoon, everybody, and thank you for giving me this opportunity to talk to elite university students. I'm sure the first question is, how come you are a neonatologist and what do you have to do with this new subject? There are several reasons for that, and I will mention two of them. The first one is that neonatology was one of the first fields in which the microbiome products were used to reduce the incidence of a common disease in ICU, which is necrotizing enterocolitis, which has a very high mortality. And more than a decade now, we have been using the probiotics, which is a product of the microbiome, to reduce the incidence of this killing disease. The second issue, I had myself an experience with it. I had a GI problem a few years back, and I used the products, and thank God it helped me to be cured from this problem. For that reason, I started reading. Unfortunately, there are no ways that we do.
Dr. Ibrahim. Okay, so I'm having a bit of a connection issue. Is it from our side? This side? Okay, it's from our side. That's nice.
Can you hear me?
Yeah, doctor, we disconnected because of an internet. Can you read what you're saying about the patient who came to you?
Okay. Shall I go from the start or just continue?
Yeah, you can continue from when the patient. Yeah.
So two main reasons that I'm interested in this subject, that neonatology was one of the fields in which the products of the human microbiomes are used clinically. We are using probiotics to prevent or reduce the incidence of necrotizing enterocolitis. I had also a personal experience with it. So for the last 4 to 5 years, I've been into this field with interest along with my colleagues, and we are looking forward to get going on this field and encouraging our colleagues. I've been lecturing in several meetings and different specialties in this field, and I'm very happy to talk to you today. Try to be as brief as possible because the topic is so huge. And I will be trying to, I will introduce the history of the microbiome, the Human Microbiome Project, how is it studied, some of the definitions, and what has been known, and what does the future holds for us. It may not go in that order, but I will try to cover it in a brief way. The human microbiome word or term was coined by a Nobel Prize winner called Nobel Lederberg in 2001. And since then, the name is used to describe what we have been knowing in the past 3 or 4 decades as the commensal bacteria or commensal microbes which lives with us. In the West, since there has been a significant reduction in incidence of infectious diseases and other causes of mortality, and there's a significant rise in chronic diseases. So most of the developing countries, since the end of the human genome project, there was a shift into studying what is around us from microbes, bacteria, etc. And more evidence started coming up to indicate that the bacteria and viruses and fungi we are carrying or living with is probably a very important cause behind the chronic diseases. So the Human Microbiome Project was started in 2007 by most of the developing countries. And one example of that in USA in 2007, one part of the Obama healthcare was to support this Human Microbiome Project by more than $200 million. And that was done the same in most of the developing countries. And since then, you can see clearly here that the publications in this field has jumped significantly, both in the gut microbiome studies and the total microbiome studies, reaching now actually in the range of 10 to 15,000 publications per year. Unfortunately, we are far behind, and I had came across very, very few references or articles talking about microbiome in Saudi Arabia. So what is the microbiome? This slide is a summary introduction to what is it actually. First thing I could say, or the easiest thing is, we can say that for every human gene in your body, there are 360 microbial genes of these microbiomes. So this is us, and this is what is inside or over our body. It's a huge number of genes, and for a 70 kilogram person, there are about 2 kilograms of microbiome in his or her body. It consists of 100 trillion bacteria and archaea, 500 trillion viruses, plus fungi, and so on. It produces thousands of chemical metabolites, and it forms the main driver of our immune system and control of digestion. So that's a brief summary of what is the human microbiome. How is it studied? Since the 16th century, they have been looking at the microbes under the microscope with limited actual information. Then the culture came, and it's only, as I said, since when the geneticists finished with the human genome studies, they started looking at the genes of these microbes. So it's only from probably the 18th century and so on. As I said before, we know them as bad bacteria or pathogens, and good bacteria, the commensals, but the terminology has changed since we know how huge and how big is the human microbiome, and that there's more and more informations are gathered after the start of the Human Microbiome Project worldwide. There are two main studies or two main procedures or ways of studying the microbiome. The first one is the 16 ribosomal RNA gene, which is a very specific and special for bacteria. And from that, they were able to look at most of the bacteria. The 16 ribosomal RNA is looked at by PCR and sequencing, and the bacteria was identified. That gave us, or gave the scientists some knowledge on the bacteria or the components of the microbiome. But recently, the shotgun metagenomics developed further and provided an enhanced view of the kingdom and species of the microbiome, where the whole DNA is taken, it's fragmented and sequenced, and by that they were able to identify more and more of these microbes in our body or in every organ. The different kingdoms were identified, the taxonomic identification of the strains and so on. And furthermore, they were able to look into the gene functions and metabolic pathways enrichment by these microbes. So to summarize it more, the 16 sRNA was the first step, then the shotgun metagenomics, and further on, there were more and more studies on what does these microbes produce and how they interact with each other and how they interact with the host. So there is further studies of the RNA, looking at the gene expression. There was more metabolic studies, and they were able to identify significant metabolic products of these microbes. This is the tree of life, and there are more than 50 known bacterial phyla, but only 6 of them were found in the human gut. And by the way, here I must mention that 80 to 90% of the human microbiome resides in the gut. So there are generally 6 phyla found in the gut: Bacteroides, Firmicutes, Actinobacteria, Proteobacteria, and so on. Again, this is if we look at the gene size, this is the human gene, and this is the human microbiome gene. So it is very huge compared to our genome. Of course, the difference in size, the human to its human microbiome, is because our cells are bigger than these microbes cells. Otherwise, they are much, much bigger than us from genetic components point of view. Again, it's bacteria, fungi, viruses, and other components, but more studies and more concentration has been in the studies has been focusing on the bacteria, and there are more and more studies looking into the viruses and the fungi. The microbiome in our body exists nearly everywhere. The majority is in the gut. Second organ is the skin, and then it comes in different types and concentrations in the mouth, the pharynx, the respiratory system, the urogenital tract. And in each part of the gut, there are specific and special microbiome, and they actually, the colon forms the most important part of the gut in which the microbiome resides and interacts with us. Microbiome also present in breast milk and also in the placenta, which previously it was thought that the placenta and the uterine cavity during pregnancy is sterile, but there is more evidence that our microbiome starts to develop with us in utero. Here's the electron microscope picture of looking at a small piece of the gut. And you can here see these are the microbes, collection of microbes, huge, separated by a layer of mucus from the cells, the epithelium of the gut. And here's the virus patches and the lymphatic system and neurotic system of the gut. So I hope this is a clear picture of the microbiome, or the bacteria and viruses and what's on, which compose the microbiome, separated from the human epithelial cells by a layer of mucus, which is very, very important in the function and of the microbiome. Then these are the luminar cells, and below it is the virus patches mainly, or the lymphatic system and neurotic system in the gut. Another picture showing the colonic mucosa, the mucus layer. These are the microbes below it, or inside the organ. Another picture showing it much more beautifully You can see the gut mucosa. This is mucus, which is not shown here, it's black. And these are the microbes or microbiome. What does it do to us actually? This is a simplification of the interaction between the microbiome and the epithelial cells, the host cells, and the lymphatic and neurotic system. There's a great and interesting interactions there. A little bit more delineated picture, but don't worry about the details. What you should actually know about this is that if this is the different microbes forming the microbiome, it has a very significant interaction with the gut epithelial cells separated from it by mucus layer. This mucus layer is interestingly and clearly shown now that it's produced by certain types of the microbiome. And its absence causes a lot of problem and a lot of damage to the epithelial cells. And I will talk more about the leaky gut syndrome. The epithelial cells of the gut, there are very tight junctions between them, but the chemical materials produced, or the metabolites produced by the microbiome, interacts with what is below these epithelial cells through these junctions. It passes easily, and it maintains the tightness of these junctions, preventing some of the bugs and microbiomes to translocate and travel inside and cause more and more disturbance. So the most important interesting interactions between the microbiome occurs mainly in the gut. It is separated from the gut by a mucus layer to prevent direct invasion, but its metabolites passes between the junctions or through the junctions between the epithelial cells and interact with the immune system, priming it from in utero, as I said, and in early days and months of life. And it interacts also with the enteric nervous system. So again, in summary, these are the epithelial cells and these are the microbiota or microbiome. They produce metabolites. The two most important metabolites produced by the microbiome are acetate and butyrate, but there are more like propionate, indole, and peptides, polysaccharides, and so on. And as I said, it interacts with the immune cells below the epithelial cells, in which 70% of our lymphatic system is in the lining of the gut. So you can see the importance of this interaction between the human microbiome and lymphatic system to prime it and to train it. And later on, it may unfortunately cause most of the problems with the chronic diseases. So it interacts with the immune system, the fibroblasts, the enteric neurotic systems. These interactions can produce either pro-inflammatory when there is complete healthy microbiome. When it is disturbed, what we call dysbiosis, when there are changes in the human microbiome, and anti-inflammatory products are released, causing the chronic diseases. The three most important organs affected by the microbiome or regulated or interacts with the microbiome, as we said, is the lymphatic system, the metabolic system, and the neurological system. One thing I must mention here, the diet is the most important material or most important product which regulates our microbiome. If you are eating a healthy diet, your microbiome will be healthy. If you are eating a Western diet or junk foods, your microbiome will be unfortunately disturbed. And this has been shown in several studies looking at vegetarians, versus Western food versus other type of foods, the more fiber you eat, the more it is, and this is one of the main functions of the microbiome. It degrades the dietary fibers and use it as a prebiotics for its growth and its differentiation. And after that, it produces from it the metabolites which interacts with our systems and leads into healthy human being or diseased human being. To summarize the gut microbiome, fine balance between what does the pathogens do as compared to the common cells. The common cells of the human microbiome inhibits pathogen growth, converts raw drugs to active metabolites, so it even helps with our medication consumption. It degrades the polysaccharides of plant origin, it produces folate and vitamin K, it produces short-chain fatty acid, which I already mentioned on their importance, and so on. While the pathogens, we know that it leads to sepsis, infection, inflammation, and damage of the organs. So there are protective functions against pathogenic bacteria in this microbiome. It interacts with our immune system and leads to development of a normal immunological system or abnormal system leading to chronic diseases. It is very important in digestion and metabolic functions, and it has a very great role in our neural development. So, what does the gut microbiome disease or dysbiosis or perturbation do to us? When your microbiome becomes disturbed, there is a wide range of diseases has been shown to have a clear association with disturbed microbiome. And you can look at every chronic disease, it is there, starting from diabetes, obesity, cancer, depression, autism, and so on. And there's a term which has been actually being looked at by the microbiome scientists, is that probably the significant disturbance in the microbiome leads to leaking of the gut, disturbance of the junction between epithelial cells, and leakage of abnormal cells, and even translocation of certain bacteria from this microbiome to invade other organs and cause the chronic diseases. The microbiome, as I said, develops with us from in utero. Birth is very, very important. Start of the microbiome health. Babies born by cesarean section are different from babies born vaginally. But fortunately, this disturbance does not last long, especially if the baby is breastfed. And here on the growth and development of the microbiome goes on with us. And it reaches actually the first 3 years of life are very, very important in directing the growth and the health of the microbiome. If we start feeding normally, then we'll have a normal microbiome. And the components and type and numbers and so on of the microbiome of baby is not like that of a 1-year-old or so, but they reach the adult life about 2 to 3 years. And then even during the adult life, the microbiome change along with us by different types of bacteria, which are known in each age and in each disease. You don't need to bother yourself about the types at the moment. I cannot skip going into a little bit of neonatology. As I said, the microbiome of the baby develops in utero, so a healthy mother will have a healthy baby's microbiome. Obese mother may lead to disturbed neonatal microbiome, and later on the baby may become unhealthy with certain diseases. As I said, vaginal delivery is more healthier and normal microbiome will develop after that, while cesarean section may disturb the microbiome. Preterm delivery is a major disturbance to the microbiome, and that's why probably the neonatology is one of the fields in which the microbiome was first studied even more than 20 years now. Actually, we have known that the microbiome, an abnormal microbiome in premature babies lead to the killing disease, what we know as necrotizing enterocolitis, where the gut becomes necrotic and most of the babies would die because the mortality is very, very high. Thank God that now we are using probiotics in neonates, and the incidence has been reduced by more than 50%. How, well, actually, the best way microbiome has been studied is by using the germ-free mice. This vessel has been used to study many diseases, but it has a special interest in microbiome. And one classical example, which has been repeatedly shown in several studies, that when the fecal microbiome of an obese person is introduced to the germ-free mice, it develops obesity. While if the fecal microbiome of a lean person or a thin person is introduced to the germ-free mice, it will keep a normal weight. And this is what is called known now even in humans as the fecal microbiome transplant, and that has been used in kwashiorkor, or actually studied in kwashiorkor, normal and kwashiorkor baby fecal material given to or introduced by enema to the germ-free mice. The disease was produced by the fecal microbiome of the diseased infant. There are a lot of work in autism, and it may hold a promising future for reducing the signs and symptoms of autistic children. Fecal microbiome transplant is an important field, has been going on again for more than 10 years now, and one of the major successes is the reduction or the cure of Clostridium difficile infection. When antibiotics is used, it has no comparison to the results when the human microbiome from a healthy human being is given through enema to the people who have this pseudomembranous enterocolitis because of the Clostridium difficile, and the success rate was 90%. As I said, it's been used now to treat autism. It did not show much improvement in the ulcerative colitis and inflammatory bowel disease, but there is more progress in that. How the microbiome is given now, it's actually the probiotics is the main product of the microbiome, which is available commercially. These are live microorganisms which are either added to food or given as supplements. Prebiotics are the compounds that promote the growth of and the activity of these beneficial microorganisms. Commonly, the probiotics and prebiotics are used together in capsules or other forms of medications, and this is called symbiotics. And they are available in capsules, tablets, solutions, creams, and whatsoever. Something very interesting about the microbiome, it is more accurate and more differentiating in us than the thumbprint. There's a very unique signature of human microbiome for every and each one of us, which is different from the others, even in identical twins. So maybe it holds more future in more than medicine. So what is my message to you today is that the human microbiome is an evolving medical field which has shed light into the origin of chronic diseases. It holds a promise with ongoing research to open a frontier in the prevention and cure of chronic diseases. It is forming the basis for personalized and precision medicine. As I said, for each one of us, there is a unique microbiome. From the perinatal till the end of life, microbiome plays a major role in upregulating our immunity, the metabolic function, neurological function, and other functions. And sorry if I have taken a long time with you. Thank you very much.